Right. Good morning, everyone. I hope you had a good week off and you had some time to rest and recharge. Here we are for the second half of the course. We are just 7 more weeks away from finishing the semester. So, as I told you at the beginning of the course, the second half is independent of the first half. So everything that was tested on the first test will no longer be tested in the second half, right? So we're in a way starting brand new. We also have 5 lessons in the second half, and the format is pretty much the same as the first half. You're gonna have 2 quizzes, 10% each, and then you're gonna have another lab, as well as the final test. Final test is being 20% and the lab is 10%, okay? Yeah, it's a little bit more difficult compared to the first half because there are a lot more application and problem solving in the second half. It's not just raw memorization. So please try not to fall behind too much, and do ask questions if you need extra help. So today we will start with lecture number 6, which is cellular reproduction. And we do have quite a bit of note-taking for the day. So please make sure you have, your pencil, crayons, and paper and stuff like that. So let's get started. So this lesson is all about how cells divide. We know from the cell theory, one part of it states that all cells come from the division of pre-existing cells. So if you have a cell here, and you need to get more cells, you would have to split the cell, and you would end up having 2 new cells. Okay, so we also talked about how much genetic material we have in a human cells. Does anybody remember how many chromosomes do we have? 46, that's right. So you have 46 chromosomes, and before you divide, you actually have to do DNA replication. You have to copy all your genetic materials so that when you do divide, in the end, each cell will also get 46 chromosomes. Okay. Now, because of things we're going to talk about later, 46, when you copy all your DNA, like you would think it's 92 afterwards, right? Because 46 times 2 is 92. But because of the way we count them, it's not quite 92, okay? It's still 46 actually. But don't worry about it. That's why I leave it blank. Okay, don't put any number here. I'll explain it to you later on. But the point is, you got to copy all the genetic material before you separate the cells. And that's what this lesson is about. It's separated into 2 halves, okay? Today, hopefully, we'll be able to finish talking about how DNA replicates. And then next class, we'll talk about the actual cell division. All right? So here in this picture, these are actually viruses. Okay, that's what they look like. Not all viruses look like that, but this particular one, it's called a bacteriophage, and they infect bacteria, which is what this brownish looking thing is at the bottom of the screen is. So let me turn off the light to show you. I'll turn it back on in a bit, okay? But you see the virus here, there is something that's being injected into the bacteria, okay? So that piece of thing that's being injected into the bacteria is what will hijack the bacteria and then start to make more virus particles. Okay, so then the bacteria essentially become a virus factory, and it's going to create new viral parts, which will then be released from the cell. Okay? So that piece of thing that's being injected into the bacteria is something that we're going to look at in a bit. All right, so first we'll start the lesson with some brief history in terms of discovering DNA. being the hereditary material. We'll talk about two important experiments, how they were done, and the purpose and the contribution of those experiments. Like I said, lots of note taking today. So in the past, people as in like scientists, right? They were like separated into two camps, right? One side suggested that DNA is the hereditary material. When I say hereditary material, I mean it is information that's being passed on to the next generation, right? When you reproduce, right? So some people say it's DNA that's responsible for heredity. Others say it is proteins, right? Because protein is made up of 20 different amino acids, right? Whereas DNA is only made up of 4 nucleotides. So if you think about it, it seems proteins would be able to encode more information just because it has more variety in its structure compared to DNA. You guys understand that? Right? You're able to hold more information with 20 different things, building blocks, as opposed to just 4, right? So that's why some people believe it's the protein that's the hereditary material. Okay, so there are two sides, and people couldn't really, figure out. And then later on, there was a famous experiment done by these two scientists, and it's called the Hershey and Chase experiment. Okay, the Hershey and Chase experiment definitively shows that DNA is the hereditary material and not proteins. Right? So we're gonna take a note and talk about the Hershey and Chase experiment. Right? So please take out a piece of paper, and then we're gonna do this. Hershey and Chase experiment. Purpose. To determine whether protein or DNA is the hereditary material. Again, hereditary means pass on to the next generation. So is it protein or is it DNA? That's what they're trying to figure out. So in their experiment, they use a type of virus called the bacteriophage. So I'm just going to draw a bacteriophage for you. I showed you a picture of them earlier, right? So they actually do look like this. So this is a bacteriophage. Which is a virus that infects bacteria. It's made up of two things. There is a protein coat and then there is a DNA core. Now, this next part is important to understand. All right, so maybe just pause for a second, listen to what I have to say, and then continue to copy. At the time, people understood how virus work. Okay? They know for the virus to make copies of themselves, the virus has to attach to its host, in this case, the bacteria. Okay? Something is injected into the bacteria, and then that something is used to make more viruses. Okay? What they did not know was what's being injected into the bacteria. Okay, like is it the protein that's get injected, or is it the DNA that gets injected? So this experiment is going to go answer that question, because whatever was injected into bacteria, that would be the molecule for heredity, because that's the stuff that the virus used to make more viruses, right? You guys understand that? So they know something was injected. They just don't know what it was. Okay? So the protein coat is on the outside of the virus, and it's basically packaging some DNA in the inside. So something from the virus was injected. So that's what they are trying to figure out. What got injected into the bacteria? Bacterium is singular, bacteria is plural. So Hershey and Chase, they grew two different batches of viruses. So batch one was grown in sulfur 35. Did you guys learn about isotopes in chemistry? Actually, I taught you isotopes, I think, right? So sulfur, if you look at the periodic table, it's usually 32. This one is heavier, sulfur, okay? Sulfur 35, which actually is radioactive. Okay? So if you put radioactive atoms in an experiment, it gives you a way to keep track of them. Okay? So please please try to understand this. We have the virus. The outside is made up of what? Proteins. The inside is the DNA, right? So if you grow viruses in radioactive sulfur, is the protein going to be radioactive, or is the DNA going to be radioactive? What do you guys think? Protein or DNA? You already forgot everything from test one, eh? Which molecule has sulfur? Does DNA have sulfur, or does protein have sulfur? Proteins, right? Protein has trace of sulfur, right? So if you grow the virus in radioactive sulfur, only the proteins will be labeled, okay? So in this case, we're going to draw the viruses like this. And then I'm going to use red to represent the radioactivity, okay? Oh, sorry. I mean sulfur, right? I went ahead of myself. So the protein is going to be radioactive, but the DNA is not.
Batch 2 was grown in. Can you give me atom to label? Something that's only found in DNA, but not protein. Phosphorus, excellent. Okay, so it's going to be grown in 30, phosphorus 32. And so again, this is a radioactive um, isotope of phosphorus. And phosphorus, if you look at the periodic table, is usually 31. This one is 32, so it's heavier. It's easy to break down. It's radioactive, okay? So these viruses, they are going to have radioactive DNA. Again, when I say radioactive, like it's some people think it's like, good or bad. It's nothing to do good or bad, okay? In the context of this experiment, when you label something radioactively, it just means you can keep track of it. Okay, you can detect where the radioactivity is afterwards, and it allows you to know which part of the virus goes where. Any questions so far before I tell you more?
If you go have like a CT or MRI, they inject you with radioactive stuff.
That's right. So glow on the image, right?
Yeah.
Exactly. Okay. So there are two more things you have to understand before I can tell you more, and try to understand it. Okay. If you don't, let me know, and I'll try to explain it until you get it. Okay. Because if you don't get this thing, everything will not make sense. Okay. Bacteria and viruses, which one is heavier? Bacteria and virus, which one would be heavier? Which is bigger and heavier? I mean, bigger is not always heavier, but then, like, which one is heavier? Bacteria is heavier, right? Okay, because bacteria is like so much bigger in size compared to the virus, so it's heavier. Everybody okay with that? All right. So, they have a test tube. Just maybe watch first and then understand before you draw it. Okay, so this is a test tube. Okay, like the ones you use for your experiment. Right. So the test tube is filled up with um the mixture, right? So they let the virus infect the bacteria, and then they put this mixture with the bacteria and the viruses into a test tube, right? And then they take the test tube and they spin it at really high speed. Okay, this is called centrifuge. Okay, you might have seen it on TV where there's the machine that spin things at really, really high speed. And when you spin things at really, really high speed, heavier stuff is going to sink to the bottom. Okay, and lighter stuff is going to be remaining at the top. It's not difficult to understand. I think of salad dressing. You let it sit there in the fridge. Next time you take it out, like the heaviest stuff, like the chunky stuff, right, all goes to the bottom, right? It's a similar idea. Centrifugation just makes it faster. Okay, the process faster. Okay, so having said that, they let the virus infect the bacteria and then they spin it down. Do you expect the bacteria to be at the top or at the bottom based on what we just discussed? Exactly. Okay. So, number one, virus infects bacteria. Number two, separates. Again, centrifugation, to centrifuge something is just to spin it at really high speed. So remember, earlier I was telling you the virus injected something into the bacteria, right? And then you go separate it. So the bacteria sinks to the bottom. So this bottom portion is basically bacteria plus whatever was injected into them by the virus. Okay, because it's now part of the bacteria, right? Whether it was the protein that got injected or the DNA that gets injected, now is part of the bacteria, so it's going to sink to the bottom. And the top here is just the virus. Part of the virus. That was not injected. Now, they did not know, right? I keep on saying they didn't know what got injected into the bacteria. Do you know what got injected into the bacteria? Do you know which is the hereditary molecule? Is it protein or DNA? Yeah, DNA is the hereditary material, right? We all know that, right? Okay, I really hope you do. So spend like first half of the course talking about that. So it's the DNA that got injected, right? Keep that in mind. They didn't know. They're trying to find out. So here's the result. Okay, result. Batch 1. Okay, so look at this. Batch 1 is the viruses that have the protein that's being labeled. They use batch 1 to infect the bacteria. They spin it down. They separate it with centrifugation. So do you expect the radioactivity to be at the top or at the bottom? Let's think about everything that you know from the batch 1. Do you expect to be number 1 at the top or number 2 at the bottom? Show me what you think. Sure. Batch 1 was labeled with the proteins are labeled, right? The proteins are radioactive. They infect the bacteria with these viruses. Okay. So after these virus inject something into bacteria, they separate it. So the virus goes on the top, and then the bacteria with whatever was injected goes to the bottom. Okay. The protein is radioactive. So I'm asking you, what result do you expect? Do you expect the radioactivity to be at the top or at the bottom? In other words, did the protein went into the bacteria? No, right? Okay. We know proteins don't, it's not hereditary, right? Okay, and that's what they found. Okay? They found the radioactivity only detected at the top portion. What does this mean? Therefore, protein coat of the virus did not go into the bacteria. You have to understand this part. Do you know why this result would lead to this conclusion? If the protein went into the bacteria, you would have detected the radioactivity at the bottom portion, right? Because the bottom portion is the bacteria plus whatever was injected into it. Okay, they did not detect any radioactivity at the bottom, so the protein did not go in. Make sense? Now you got to understand, this only shows the protein didn't go in. It does not actually show the DNA went in. Because there's no way for us to know where the DNA is. Maybe the DNA stayed outside too. Okay, which is why they needed to do the second half of the experiment with batch number 2. So batch number 2, only the DNA are labeled. And this time, they found the radioactivity at the bottom. So based on the results, they concluded that DNA and not protein is the molecule for heredity. Okay. Here, I'll make it bigger. Okay, does anybody have any question? Which word are you looking at, Jenny? Over here? For heredity. Sorry, heredity. Okay, so this is the Hershey and Chase experiment, all right? I'm not gonna ask you to describe it on a quiz or whatever, obviously, but you have to understand the process in order to answer some of the questions, okay? All right, moving on. This is basically what it looked like visually. Over here, we have the batch that's labeled with the radioactive sulfur. So the protein code is red. And then the DNA was being injected. And you can see the protein code remains outside of the cell, which is why we saw the radioactivity only at the top portion. However, with batch number 2, when you label the DNA went into the bacteria, and bacteria being heavier, it's going to sink to the bottom when you spin it down, and they detected the radioactivity at the bottom portion only. Okay. All right, so now people know that it was DNA that's responsible for heredity. Then the next thing they were trying to figure out would be the structure of DNA. Okay? So there are a couple people who contributed to that effort. There was Rosalind Franklin, which she used X-ray to study the structure of DNA. and she actually was credited with the first to be the first person to figure out that DNA is a double helix. Okay, you know DNA is twisted, right, like a ladder, so like double helix. But then she died of cancer before she could finish her work because she was working with X-ray a lot, and the protective measures were not particularly in place back then, I suppose. And so Watson and Craig, which is these two people, they took her data and continued to work on it and ultimately figure out the three-dimensional structure of DNA. Okay, so they were awarded with the Nobel Prize for doing that. But people nowadays say, they should have credit Rosalind Franklin as well, because it was actually her data that allowed them to created the 3D model. Okay, so this is the X-ray data from Rosalind's experiment, and then in 1953, Watson and Crick built the first scale model of the DNA double helix. All right, now people knew what DNA looked like, the double helix twisted together. The next big thing to try to figure out was how does the body make copies of DNA? Okay, so like I said, we have 46 chromosomes. Chromosomes are basically condensed version of DNA, right? And every time we need to copy ourselves, we need to copy the DNA. So how was it done? Well, there were three proposed model at the time of how this actually happens. And these people, Messelson and Stahl, they did an experiment to show that which of the three models was correct. All right, so let's just go back a little bit first here. The purpose of DNA replication, why do we need to copy the DNA anyways? Well, as I told you at the beginning of today, before a cell divides, it must copy all of its DNA so that each daughter cell will get the complete set. Okay, so this is called a parent cell, not like your parents, okay, but like a cell inside you that is about to divide. That cell is called parent cell. After that cell divides, then you get two daughter cells. Okay, so you can see it started with two chromosomes here, the blue and the red, and then you make a copy of each. So now you have two blue stuck together and two red stuck together, and then you separate them. So each of the daughter cell will have the very same exact copy as the original. So that's why we have to do DNA replication. There were three proposed model of DNA replication. We have semi-conservative on the left, conservative in the middle, and dispersive on the right. Okay, it's a little bit difficult to understand from this. So hopefully this picture is going to help you. Took me half an hour to make this, so it better helps you understand. This is the original picture. And over here, when you make a copy, we have the original, and then we have the copy. This is pretty much like how photocopier works, right? You get the original exactly as it is, and then the new one that's made up of just the copy. So this version of copying is called conservative. Okay? Conservative. You preserve the original entirely. In another model, you make a copy, but you basically take the original and you rip it in half, and then you build a mirror image of the original. And so this is original, and then you just build a mirror image. And then this is original, and then you build the mirror image. So this is semi-conservative. You are preserving half of the original material, and then the new half is built using the original half. So we call this semi-conservative. The last model is called dispersive. So rather than, it's kind of like semi-conservative. However, when you preserve the original, you are not preserving the entire left half. You're still preserving half of it, but the half is kind of scattered all over the place. Okay, it's like a patchwork. So the end result is going to be a mixed match of new and old materials scattered throughout the entire thing. Okay, dispersive. Do you understand the three different models? All right. Let's take a look at the DNA picture again. Pink here represents the original DNA. In semi-conservative, you would separate the two DNA strands. Remember, DNA has two strands, right? They are complementary. If you know half, you can predict the other half, right? If it's AAC on one side, what is it on the other side? AAC on one side, what's on the other side? TTG, right? Okay, so knowing one half allows you to build a mirror image, right? So this is the original, the parental DNA. You separate it, and then you build a new strand. The blue represents the new. Make sense? Semi-conservative. Conservative is you keep the original, and then you copy it. Dispersive, you are not like really separating the strand like semi-conservative, but like you take bits and pieces of it, and then you build bits and pieces of it. And in the end, it's like a mix of old and new all over the place, right? Do you understand the three models and how the picture is related? Huh? Now that's the first division. Cells don't just divide once. Right? One cell can divide and become two. Each of those cells can divide again and become four, right? So let's say we follow another round of division. If we follow the semi-conservative model, we will separate the blue and the red, and then we will build another strand using, blue as in like new materials. It's gonna look like this. Blue. Now we are making a copy of a copy, right? That's why it's blue and blue. And then over here, the original, and then you make a copy. Dispersive again, it's just mixing. Okay. So those were the three proposed model of DNA replication. And there was an experiment to figure out which one was correct. Okay. And that is the Michelson and Stahl experiment. So we're gonna write that down. We're gonna take a note on that. Okay. Michelson and Stahl experiment. You will need ideally 3 colors for this. If you have red and blue, definitely red and blue. And if you have purple, it would be great. But I don't think anyone has purple. It's very unlikely. Unless you win one of those pens that I gave out last time. Okay, purpose. What did I say the purpose of their experiment was again? What are they trying to determine? To determine what? Yeah, I hear a lot of whisper and I read some lips. I think you guys are saying to determine the correct model of DNA replication. Is it conservative? Is it semi-conservative? Or is it dispersive, right? That's what they're trying to figure out. So, this is what they did. Right? Number one, they, as in Meselson and Stahl, grew E. coli. E. coli like the bacteria, right? E. coli in N15. Heavy nitrogen. What's the normal mass? What's the common mass of nitrogen? Do you guys remember from chemistry? You guys learn like molecular mass yet, or yeah? Yeah. Nitrogen, normal nitrogen. What is it on the periodic table? Is right there. People close to the wall, can you tell me what it is? 7 is the atomic mass. What is it? Tell me. 14. Yeah, 14. Okay. 14 is the common isotope, right? Okay. 14 is the normal one.
So N15 is heavy. It has an extra neutron, right?
Yeah. Okay. So they grew E. coli in heavy nitrogen for several generations. Okay. I'm gonna put heavy nitrogen in red, okay? For several generations, this will this caused, I guess. Because nitrogen is found in DNA, right? So all the DNA will be heavy. Next, they transfer the E. coli to regular nitrogen. You grow bacteria in a little dish, okay? So in the dish, you can supply whatever nutrients you think is appropriate. So in this case, they have dishes with regular nitrogen in them. So they transfer the E. coli to regular nitrogen. The E. coli was allowed to divide once. And their DNA were extracted and separated by weight. Okay, I'm gonna have a side note here. How do we separate things by weight? What did we learn this morning? Yeah, we spin it down, right? Yeah, we spin it down with centrifugation. So there's going to be a test tube. Okay, now this test tube is gonna be filled with some, it's kind of like a gel stuff, okay? Don't worry too much about it. Okay, there's gonna be some gooey stuff in here, and then we will separate the DNA based on how heavy it is. Okay. So we have DNA that's made up of heavy nitrogen, and then we have DNA that made up of regular nitrogen. When you spin it down, where do you expect to find the DNA that's made up of N15? At the top, at the middle, or at the bottom? Okay, anyone else? If you grow the E. coli in heavy nitrogen N15, top, middle, or bottom? I need to see it to make sure you understand before we move on. Top, middle, or bottom? Okay, yeah. Be sure of yourself. Yeah. Anyone else? Yeah. So at the bottom, I didn't see you wrong. I just want to make sure.
Oh, no, I was like,
Yeah. So at the bottom, right? Because it's heavy, right? You understand that? This is not a trick question, guys. Okay. So you would expect to find a heavy DNA here. Okay. That's like if both strands are made of heavy nitrogen. Okay. N15, N15, right? Both strands. How about DNA that's made up of regular nitrogen, N14, N14? Where do you expect to find it? Yeah, let's say top, okay? Top. Yeah. Hold on. Okay, top. Okay. Right here. Okay. N14, N14. Now, what's the middle for then? What do you think? All right, let's go back to this picture. Let's take a look here. It will help you hopefully understand that. Okay, so for example, where do you find this DNA? Top or bottom? It's the same color, guys. This is red, right? This is heavy nitrogen, N15. When you spin this down, do you expect to find it top or bottom? Bottom, right. Very good, okay? Top or bottom? Top or bottom? Bottom. Okay, what about this guy? Top or bottom? Middle, that's right. Because it's not as heavy as 1515, but it's not as light as 1414, right? One is made up of heavy nitrogen, 15. The other is made up of regular nitrogen, 14. So if you combine the mass, it's somewhere between the top and the bottom. Do you understand? This is critical, all right? So middle, right? Okay, stay with me, stay with me. How about this one? Top, middle, or bottom? Also middle, right? Okay. So if we, listen carefully, if we see a band of DNA in the middle, we actually do not know whether it's this one or is this one. All we know is it contains a mix of 14 and 15, right? Okay. Let's go back to our note here. So that's where the purple comes in, okay? If you don't have purple, just mix red and blue together. So this is going to be half 15. Again, I don't know which half. Maybe it's the entire thing. Maybe it's here and there. Okay. But it's going to be like this. Or like this. Okay. You get the idea? It's like, dispersive, right? Okay, so I'm going to show you the result of their experiment after one division. Okay, after one division. only a middle band was observed. Only a middle band was observed. Now, I want you to think about this before you answer me. Okay? I'm going to come back to this in a bit, okay? I'm just going to show you this picture again. Okay.
This is the original, right?
15, 15, 15. They're all heavy because it was growing in 15 for some time.
But now after you transfer to the 14 and we let it divide for one generation, these are the different outcomes for the 3 different models, right? So they did the experiment, they extracted the DNA, and they only saw a middle band. Based on this result, which model is definitely wrong? Remember, we're not looking at the second generation, just look at the first generation right here. Okay? Which model is definitely wrong? A, B, or C? Very good, good, good. What is that? Yeah, okay, good, good. Okay, a lot of you got it, okay? This is definitely wrong. Because if this is the correct model and you extract the DNA from the first division, how many bands do you expect to see? 2 bands. You would see a blue one, 15, 15 at the 14, 14 at the top, and a heavy one, 15, 15 at the bottom, right? But they didn't. They only saw 1 band. That's in the middle. Telling you it's either this or that. Okay?
Now, I'll draw it out for you to help you understand more. Maybe I'll copy this. See if I can do it. Give me a second. Okay, so we'll say this is the expected result. Expected result for different models. Okay, so this one is semi-conservative, right? I'm just going to put semi. This one is conservative. And this one is dispersive, right? Okay, let's draw this out and see what the expected result is. Okay. For this one, where would you expect the band? We did this already. I just want to ask you again. If you extract these DNA and you spin it down, do you expect it to be top, middle, or bottom? Middle. Good. Okay. For conservative, if you spin it down, where do you expect the band? Top, middle, or bottom? Top and bottom. That's right. Top for this guy. Oh, sorry. One more time. Top for this one. That's the lighter one. And then the heavier one would be at the bottom, right? Understand? And then what about this? Where would you expect the band? Top, middle, or bottom? Middle. Because the DNA is half-half, right? Half heavy, half light. Okay. So again, after 1 division, only the middle band was observed. So that means this is wrong. Okay. Therefore, conservative model is incorrect. But we do not know if it's semi-conservative or dispersive. Right? The data does not distinguish one versus the other, right? And that is why they had to do another division. Okay, that's why they have to let the E. coli do another round of division. I'll let you copy the stuff first. Now, don't draw this, obviously, it's already in your notes. But you know, and again, I'm not gonna ask you to explain this process on the quiz. All the questions about the experiments will only be in the multiple choice. Okay. And as always, I'll give you the study guide, like the test review when we get closer to it. Okay. Hmm? For the quiz, right? Like a review, right? Yeah. I always do, right? I know, right? First day back from your week off, it's so heavy, all this material. So already I can hear your brain hurting, right? But we must continue. Okay. So what was this? This was number 3. Okay, so number 4. Okay. The result from one division did not distinguish semi-conservative and dispersive model. They allowed the E. coli to divide for a second time before extracting the DNA. Like for those of you who have a hard time visualizing this, you actually can extract DNA quite easily. And it will look like how do I describe it? It's like a cloudy thing in a solution. Yeah, like you can actually physically see it in a test tube. Right? We used to extract them from like bananas and strawberries. But strawberry got expensive and seasonal, and we were always able to get it from in time for the lab, so we don't do it anymore. But you will be able to see a little bit of like cloudy material in the test tube. And you can actually spool it up, right, and see a long thread of it. It's quite interesting. Yeah, you actually have all the material you need at home to do it. You just need a little bit of detergent and vinegar and stuff like that. It's quite easy. So anyways, they did let the E. coli divide for a second time, and then they collected the DNA. Okay, so this is the what did I say? Result after the second division. Okay. So they saw a middle band, and then they also saw a light band, top band. Top and middle band were observed. So let's see. If you follow through for another round of division, that's what the model would predict. And now they saw two bands. So is it dispersive or is it semi-conservative? Yeah. Is it one or is it three? Is it semi-conservative or dispersive? Think about it. If you collect these DNA and you separate it, which one, where will this DNA be at? Top, right? This one will be in the middle, two bands, right? Okay, top and middle. But for dispersive, all these will only be in the middle, right? Okay, so they did not see middle only, they see middle and top. That's why the semi-conservative. Okay? The correct model, therefore, is semi-conservative. Conclusion. Semi-conservative is the correct model. Sorry, let me just copy this over for your notes as well. So again, I'm just gonna draw the expected result. Okay, so that's semi, middle and top. And then this was already rejected, right? From the first division. But if you were to do it again, you would expect to have like a middle one, I'm sorry, a bottom one, and then the top. And for this one, dispersive, you would expect to have a middle one. That's all. No more experiments for today. Those are the two big ones, okay? Hershey and Chase. Hershey like the chocolate. And Mesosin and Stoll. It's time for a break. Right? Let's take 5 minutes. We'll resume at 9:40. If you Okay, let's do a poll question. Concept check. What is the answer? Not quite. Yeah, that's good. You can look at your notes, guys. You don't you don't have to do it from memory, right? Yes, thank you. Anyone else? Yeah. So correct answer is A. Okay, to label the protein coating of the virus. We drew it up. Okay. This one is hard, even harder. I'll turn up the light so you can see it better. And still see your fingers, you can just put them up.
What is If DNA replication follows conservative model, what would you expect the results to be?
A.
Okay, so look at the drawings that we had, right?
We can go back to over here. If it's conservative,
We would expect to see two bands after the first division, right?
and then if we do it for another round, we would continue to see two bands, right? Okay, so the answer is A. Okay.
Two bands, two bands.
That's what you would expect to see if it's conservative. That's what the question is asking, right? Okay. I know, I know. All right, next we will do mechanism of DNA replication in prokaryotes. Right. Do you remember the difference between prokaryotes and eukaryotes? What are we? Are we prokaryotes or eukaryotes? Yeah, we're eukaryotes because our cells have a nucleus, right? Okay. So these are DNA that you're looking at, actually. And it turns out when you copy the DNA, you would open up a bubble, and then you would copy the DNA from the bubble outward. I'll show you what I mean in a bit, right? So it's the mechanism means the method, right? The ways by which DNA replicate is the mechanism, right? And it was easier to study that in prokaryotes, in say E. coli, than this to study in like an animal cell. So that's why earlier on, all the work that was done in prokaryotes, and then later they found out that we eukaryotes actually do it in a very similar way as the prokaryotes, okay, with a few key differences, which I'll of course tell you about in a bit. All right, so DNA replication in prokaryotes. The parent molecule being the original DNA. That's what it means. Parent molecule is the original. Unwinds and two new daughter strands are built based on base pairing rule. The way that it works in general is the enzymes that are involved in this process, they cannot copy letter by letter exactly. So what I mean is this, like if they see AATC, it's not possible for them to copy it as AATC. If they see AATC, the way that it works is they would have to make TTAG. Okay? It's not going to be able to copy exactly what it sees, but rather it's going to be building the complementary strand. When they see the nucleotides, the only way they know how to build is by following the base pairing rule. Do you understand? Okay, so like if I ask you to copy this sentence, right? You would just copy the copying of DNA. You would copy letter by letter, right? Okay. That's not how it works for DNA replication. They cannot copy letter by letter. If they see AATC, they cannot copy it as AATC. They can only copy using the base pairing rule. That's why if they see AATC, they will be making TTAG. Okay? That's one thing to keep in mind. The copying of DNA is remarkable in its speed and accuracy. It's able to copy thousands of nucleotides every second. Right? So that's really, really fast, right? Right? In humans, we have billions of nucleotides. So it makes sense to do it quite fast. Right? But speed is not the only thing. It has to be accurate. If you make a mistake, then you introduce errors in your DNA. You introduce mutations, which could cause a lot of things to go wrong. More than a dozen enzymes and other proteins participate in DNA replication. So here are all the enzymes that we'll be learning. We probably won't finish it today. Okay, we'll see how far we go. Okay, so now we're gonna take a note. This is a big one. Okay, this one is big. You probably need a new piece of paper. let me show you. Let me see if I have an old one from like last semester or something. Yeah, this is what we drawing, okay? We're drawing this big thing, and then we're gonna write a bunch of other things. Okay. So please plan the space accordingly. I would suggest landscape, okay? And then you just draw it like this, okay? All right, here we go. You know, later on in the semester, when you have a chance to like evaluate the course, I one of the things I really want to know from you guys is like, do these drawings actually help you or not? Okay, because some people think it's so annoying to copy all these things and like, messy and not make sense, but some people aren't useful. So I always am interested in to know, right, whether this is helping or is it doing more harm, in general. Okay, would love to hear about it. So mechanism of DNA replication in prokaryotes. Okay, so we are eukaryotes, they are prokaryotes. So let's take a look at a bacterium. Okay, like a E. coli, like I said. One of the biggest difference between their DNA and ours, well, first of all, ours is in a nucleus, theirs is not. But more importantly, their DNA are circular. Circular DNA. Our DNA are linear. Okay, we have 46 chromosomes, right? Each of them is like a straight line. Theirs is like a rubber band. It loops back on itself. Okay. So like if you zoom into the DNA, it's actually, like a double helix like this. Don't draw this. Okay. I'm just trying to show you. It's actually really hard to draw. But that's the DNA, right? With the base pairs. Can you visualize that? Okay, so it just loops back on itself. And that's an important distinction. Because our DNA is linear, we actually have a big problem when it comes to DNA replication. And we're going to talk about that big problem probably next time. Now, in addition to the core circular DNA, they also have extra bits and pieces of little rings of DNA. We call them plasmid. Right? These are extra DNA for special properties. Okay? And we will talk more about these plasmid later. But basically, the plasmid will give bacteria some special abilities. For example, they might be able to stick to each other better, or they might be able to pump out antibiotics. Okay, so when one bacterium meets with another one, they could actually exchange these plasmid. It's like, hey, I am resistant to antibiotic 1, and you are resistant to antibiotic 2. Let's exchange the plasmid. So afterwards, both bacteria will be resistant to both antibiotic 1 and 2. Do you understand? And so that's why at hospital, they're so concerned about these things called superbugs. Superbugs are bacteria that has accumulated a whole library of plasmids to be resistant to a lot of antibiotics. Okay? So like you might carry bacteria on your body that is resistant to antibiotic 1, 2, 3. Okay, I might carry bacteria that's resistant to like 4, 5, 6. Okay, so I visit a clinic and then I cough on my hand and I touch the surface. Okay, and then you accidentally touch the surface and then you touch your face. Now those two bacteria can find each other. They can exchange the plasmid, and now you have bacteria that's resistant to antibiotic 1, 2, 3, 4, 5, 6. Right? And then you sneeze into the air. Someone breathes it in. They now have bacteria that cannot be treated with antibiotic 1 to 6. You understand? That's like an oversimplification, but essentially that's how superbugs are created. If you're admitted to the hospital, one of the first things they ask you is, have you previously been contracted with MRSA or VRE? Okay, these are both very prevalent strain of superbugs. Okay, you don't want them to be spreading in the hospital. Anyways, so that's that. The circular DNA. Okay, let's zoom in into the DNA here, the circular DNA, and we would have the parent strand here. Let me try to draw this out. There we go. Now, previously in I don't know, lecture 2, I guess, we learned that DNA are number one, complementary. They follow base pairing rule. And what's the other property that we learned? That they go in opposite direction. What's the word for it? Excellent. Anti-parallel, right? One is going northbound, the other is going southbound, right? Traffics are running in opposite direction, but the roads are parallel to each other, right? So we don't say north and south when we talk about DNA. Instead, we use numbers. Okay. One strand is called 5' to 3', and then the other strand is called 3' to 5'. Why are they 5 and 3? Don't worry about it. Okay. If you really want to know, see me after class, but just better to know it as it is. Okay. So these are anti-parallel. Okay. They run in opposite direction. Okay. So now I didn't write the letters in here because it would be like too complicated. But basically these are nucleotides, right? Like AAC, and then this would be like TTG. You understand that? Right? There's going to be the letters in between, right? So in order to copy the DNA, the parent DNA, what needs to happen first is you need to create a replication bubble. So you're going to open up the DNA. Oh, hey, how are you? We're going to open up the DNA. I actually don't remember what his name is. That's what that's what's going to happen next semester. If I don't see you guys and then you say hi to me, yeah, then I'd be like, who is that? I will recognize your face, but I will not remember the name. It's like short-term memory. Oh, it's all recorded on the recording as well. Oh, maybe I'll delete that part when I upload it. Anyways, so when you want to copy a DNA, you gotta open up a bubble, okay? And you might open it here, you might open it here, and that's called the origin of replication or replication bubble, right? And then it will look like this. Three prime to 5'. Okay, so this here is called the origin. Origin. Oh my goodness. Of replication. Replication bubble. Now it's winter time now, and this is a problem with my kids with their winter jackets. Okay, I don't know how. Sometimes after they zip up, the zipper just bulge open in the middle of nowhere. Okay, and this is exactly what that's like. It pops open, and then you actually put your finger in and then like open the entire zipper. Right? Can you visualize what I'm saying? And that's how DNA replication works. You create a bubble, and then you unzip it along each side. Okay, so you got to be building the new DNA strand, building it. Okay, the blue here is the new DNA strand, new DNA. The black is the original DNA. And then you will expand. You expand sideways until in the end, you end up having something like this. Right, so here you have 2 copies. 5 to 3, 3 to 5. Semi-conservative model, right? We just talked about that before the break. That's why half original, half new. Right? Half original, half new. 2 copies. Now, to help you see it better, or to understand it better, maybe we can write out the actual letters. Let's try to do it together, okay? If I have a strand here on one side, A, T, C, C, G, T, A, 5 prime to 3 prime, what number do I start up here? 5 or 3? 3. And then go ahead with the nucleotides. Right. And what number do I end with? 5 prime. Okay. So watch. You don't have to copy this. You can just watch. During DNA replication, we're going to create a bubble. And we're going to separate the 2 strands. Okay, we're going to separate the 2 strands. And then an enzyme is going to come along. And then it will build. So remember, I told you, they cannot just copy letter by letter, right? So when they see a 5 prime, it's going to be opposite, of course. So when they see an A, they will know to put a T. They follow base pairing rule. And then same thing. Essentially, you are using the nucleotides to recreate the missing half. Similarly, on the other side, you're going to be creating the other missing half. So in the end, you get 2 copies of the original. Semi-conservative, because you keep the entire half of the original, and then you build a brand new one. Okay. All right. So what we're going to do now is we're going to zoom in onto one side of the bubble. Okay, so I'm going to draw the bubble again. Just draw a small one here. Okay, we have 5 prime to 3 prime and 3 prime to 5 prime. These are just the directionality. And then we're going to zoom in into this portion. Okay, this is called the replication fork. The whole thing is a replication bubble, and then you have a replication fork on each side. Replication bubble, replication fork. And this is where you have to draw big, okay? Do you understand what I'm like zooming in onto? Like, is anybody confused with the picture yet? No? Yeah. Okay, cool. Now I'm gonna put some letters in, okay? I'm just randomly creating them. Feel free to make your own if you want. If you don't want to think, you can, of course, just copy. Now, the once you randomly make up one strand, the other side cannot be random, right? The other side has to follow the rules. Okay. And of course, the nucleotide is going to continue into the rest of the strand, but I'm not gonna put them there. So this is parent DNA, the original. We are going to learn about the enzymes and the proteins that are involved in this process. The process we're trying to learn here is DNA replication, okay, in prokaryotes, right? So this is where colors would help. So this here is the first enzyme we'll learn. This one is called a helicase. Unzips the DNA. This is exactly like the zipper, the thing that you hold on to zip and then zip your jacket.
Basically, it's already opened up, and then you can grab onto the helicase and you unzip more. Can you visualize that? Okay. You just as this moves over, you will unzip it and you open up more of the DNA. Remember, these two things used to be together, right? The T is bonded to the A with two hydrogen bonds, and these used to be together, but the helicase will unzip the DNA. Okay. On the other side of the fork, you would have another helicase, right? So you basically unzip in both directions. Is that okay? That's helicase. Now, this next part might be a little bit difficult to draw, but maybe watch me do it first, and then you decide how you want to do it. Okay, keep the 5 and 3 where it is. But DNA, as you know by now, is a double helix. So this is actually going to be like all twisted up like that, right? With the bonds and everything. I mean, this part should be twisted up as well, but then I didn't draw it because otherwise it's like too confusing, right? So what happens is when you unzip something that's twisted up like that, you actually will introduce more stress and tension into the strand. Okay, it's very easy for this to kind of knot up and tie up even more as you unzip. And if there is too much tension building up in the DNA strand, it might actually snap and break at some point. Right? So that's why we need another enzyme, which is this guy here. This is called gyrase. Okay, like I said, don't look this stuff up online, okay? Like I'm trying to keep it simple for you. If you look it up, it's gonna be like more complicated. If you don't understand something, just ask me, okay? So the gyrase is another enzyme that will help
It basically reduces tension. That's building up. In the DNA as it is being unzipped. Okay. We need to relieve the tension so that it is not going to break. Last thing, and then I'll let you go. The two halves of the DNA are attracted to each other, right? Because A wants to bind with the T and the C want to bind for the G. There is some hydrogen bond that wants to keep them together. But we don't want them to come back together, right? We need to maintain the bubble. We need to keep it open so that the enzyme can come along and make copies of it, right? So that's why we need to have another protein here to keep them apart. And they're going to be stuck here. Okay, these are called single strand binding protein. It keeps the DNA strands separated. Because if you don't keep them apart, they're just gonna snap back together and you won't be able to build anything. All right, that's it. So keep this picture, review the notes. We're gonna finish talking about this process on the Wednesday.